Hello, everyone. My name is Zenia Siddiqui, and I'm going to be talking about overcoming therapeutic challenges in glioblastoma through CRISPR-Cas9 gene editing. And I conducted this research under my mentor, Miss Alexandra Massa. This is a shortened version of my abstract, but basically, glioblastoma multiforme is an aggressive brain tumor with poor diagnosis due to its resistance to conventional therapies. So, I researched how CRISPR-Cas9 is a precise and cost-effective gene editing tool that can offer unique potential to overcome this resistance by directly targeting the genetic drivers of GBM. And I will talk about how CRISPR-based strategies could transform treatment and improve patient outcomes. So, for a background, CRISPR is classified as a grade 4 aggressive brain tumor by the World Health Organization, and it is the most common type affecting 90% of patients. It's also classified by IDH status, mutated or not mutated, and patients of glioblastoma exhibit general symptoms of headaches, seizures, and neurological decline. The nonspecific nature of these symptoms makes early diagnosis very tricky. So once these symptoms are linked to GBM, some standard treatments include surgery, chemotherapy, and radiation. So neurosurgery aims to remove the tumor, but GBM's infiltrative nature and unclear boundaries with the healthy tissue make complete removal difficult, causing recurrence to happen, um, leading to high recurrence. So after surgery, patients typically receive chemotherapy, commonly temozolomide or TMZ, which targets those rapidly dividing cells, but that also harms healthy cells, which we see a lot in chemotherapy. Um, and radiation therapies like IMRT aims to destroy tumor cells by damaging the DNA, but they carry risks such as resistance and damage to surrounding brain tissue. So even with aggressive treatment, GBM often recurs, partly due to tumor mechanisms like the MGMT enzyme that repairs the chemotherapy-induced DNA damage. So all of these limitations highlight the need for innovative approaches, and CRISPR-Cas9 gene editing offers a promising alternative. So the next one is an introduction to CRISPR. Um, CRISPR stands for clustered regularly interspaced short palindromic repeats, a DNA sequence that is commonly found in the genome of archaea and bacteria. So the organisms utilize the CRISPR-Cas system as an adaptive immune mechanism, capturing and integrating short segments of invading viral DNA into their own CRISPR locus. The integrated viral DNA serves as an immunological memory for future encounters, and scientists have utilized these mechanisms in genetic engineering using the components of CRISPR-Cas9, the Cas9 nucleus, and the guide RNA. So the Cas9 nucleus is an enzyme that functions to snip DNA at a specific site, as you can see in the image below. The guide RNA has two components to it, the CRISPR RNA and the transactivating RNA. So the cRNA region is complementary to specific target sequences, allowing for specific detection. The TRAC-rRNA region acts as a scaffold or almost like a base that the Cas9 can actually bind to. In mechanistic applications, these two components are typically engineered into a single guide RNA for simplicity. Um, so there are many mechanisms of CRISPR-Cas9, and it is these mechanisms that make it so precise. So starting off is target recognition portion. Researchers begin by designing a specific gRNA with a sequence that is complementary to the specific sequence they want to target in the genome. This gRNA is then delivered into the cell, and it binds with the Cas9 protein or the DNA or RNA that encodes the Cas9 to make an entire complex. This complex then scans to find the complementary DNA sequence in the genome, where Cas9 then functions to make a double-stranded break, a DSB, at the targeted location, utilizing its two nuclease domains, H&H and RUVC, to achieve that double-stranded break. To respond to this double-stranded break, the cell responds using one of two natural repair pathways, the non-homologous end joining NHEJ pathway or the homology-directed repair HDR pathway. So While the NHEJ pathway is quick, it is rather error prone and it often leads to small insertions or deletions, and that can affect the gene's function. The HDR pathway is a template dependent mechanism that enables precise gene correction, although the HDR pathway is longer. But in general, CRISPR is very programmable, efficient, and versatile. And its versatility is seen through its many applications. So these are just a few of the applications of CRISPR, but I'll just go through each of them. So in gene knockout, the Cas9 creates a double-stranded break at a targeted location to which the cell repairs the damage through the NHEJ repair pathway, resulting in indels that kind of disrupt the gene sequence, knocking it out by silencing its function. So you're basically removing that gene. It can also correct mutations in tumor suppressor genes, such as TP53, which is a very common cancer gene, restoring their protective role against cancer. For gene insertion, CRISPR-Cas9 can use the HDR pathway to insert new genes, and it can insert helpful genes, but most of the time it uses suicide genes that induce apoptosis of the harmful cell. In gene regulation, scientists can use modified versions of the Cas9 to switch on immune-related genes or silence the drug resistance genes like MGMT. And finally, and most importantly, CRISPR allows multiplex editing where multiple genes are targeted simultaneously. And this is very critical for glioblastoma since it involves so many overlapping genetic altercations, which is why being able to target all of them at once makes the process a lot easier and straightforward. So despite its promise, CRISPR in GBM therapy faces major challenges and it has a long way to go. Efficient delivery across the blood-brain barrier is difficult, and ensuring that CRISPR edits are precise and only the intended DNA sequence is critical to avoid harmful side effects. Tumor heterogeneity means that no single target will work for all patients, and GBM cells can actually adapt rapidly to the therapeutic pressure that scientists put on them. There's also a risk of immune reactions to Cas9 or delivery vectors, since we're basically introducing a new thing into a person's body. So for CRISPR to become a clinical reality in glioblastoma, these challenges must be addressed through further refinement and preclinical to clinical translation. However, CRISPR-Cas9 represents a paradigm shift in how we treat glioblastoma, offering genome editing that is far more precise than any conventional therapy that I mentioned before. This technology can enable personalized treatments tailored to each person's unique tumor mutations. Newer approaches like base editing, which allows single letter DNA changes without double-stranded breaks, and prime editing, which acts like a search and replace tool for DNA, are expanding CRISPR's capabilities. At the same time, researchers are developing safer delivery methods, such as nanoparticles, engineered viral vectors, and even exosomes to effectively cross the blood-brain barrier and minimize off-target effects. So while challenges do remain, these advances highlight CRISPR's potential to significantly extend survival, reduce tumor recurrence, and ultimately improve the quality of life for patients that have GBM. Thank you so much for listening. If you have any questions, please feel free to email me at the email listed over there. And here's a QR code that links to most of my sources that I have used in this presentation. Thank you.